Welcome to the deep dive. Today, we're exploring something really quite fascinating.
Yeah, it's this convergence of, well, machine learning, smart computers, and plant science.
Specifically, how scientists are using AI to understand the relationship between plants and the tiny microbes living with them.
Mhm.
We're basing this on a consultation record from a research project looking into exactly that, predicting these interactions for, smarter agriculture.
Exactly. So our mission today really is to unpack how this machine learning approach could help design, let's say, beneficial bacteria cocktails for plants.
Making them grow better, be more resilient. Sounds great. Okay, let's start with the basics. The plant microbiome. What is that?
Right. So think of it as a whole community. Microscopic organisms living in and around the plant. Roots, leaves, the whole system.
Like a hidden ecosystem.
Precisely.
Yeah.
And this community can seriously help the plant. It can boost stress tolerance, help it grab nutrients. It's like having extra tools.
Like a biological software upgrade provided by these microbes?
You could think of it that way, yeah.
Okay, so what's the main hypothesis driving this specific research project?
The core idea is that different groups, different types of microbes, trigger unique responses in the plant. They switch specific genes on or off.
Creating distinct patterns, like signals.
Exactly. These gene expression patterns are signals that, well, the AI can learn to recognize.
Which leads to the goal, right? Using these signals to train AI models, what do they hope to achieve?
The main objective is prediction, being able to predict the outcome when a plant meets certain microbes or combinations of microbes.
So you could design the perfect mix.
That's the idea. Tailored microbial treatments for specific plants, specific conditions to really boost their performance, precisely managing them.
Okay, makes sense. How do they actually do that? How do they teach the AI? What's the input data?
So they use what are called labels and features. The labels are based on differentially expressed genes, DEGs.
Okay, DEGs, that's the plant's response. Genes turning on or off.
Exactly. That pattern of on-off is the label the AI learns. The features are things like, well, which specific types of microbes are present.
Ah, like the examples in the notes, Actinomycetales and Rhizobialis.
Yes, those specific groups. They look at how the presence of those correlates with the plant's gene activity, the DEGs.
Gotcha. And the prediction method they used initially.
They started with random forest models. It's a type of machine learning good at figuring out which features, which microbes in this case, are most important for predicting the outcome.
The outcome being the gene expression in the roots and leaves.
Correct. In both roots and leaves.
Any preliminary results? Anything interesting pop up early on?
Yes, actually. One thing stood out quite clearly. The random forest models consistently flagged Actinomycetales as important.
Important for predicting the gene expression.
Yes. And what's really interesting is this was true for both the root samples and the leaf samples.
Wow, okay, so that consistency across different parts of the plant suggests they're really involved, right? Not just hanging around.
Exactly. It's a strong signal. Suggests they're active players in the plant system.
Okay, that's a key finding then. Now, the consultation record also had advice, right? Next steps for the researchers.
It did. The advice was basically to broaden the toolkit. Try other machine learning models.
Like which ones?
They specifically mentioned XGBoost, fully connected neural networks, and also LightGBM.
And the point of that is?
To compare performance. See which model gives the most accurate predictions for how specific microbes change gene expression in specific plant tissues, finding the best tool for the job, essentially.
Right, refining the prediction power. So let's zoom out. What's the big takeaway here for us, for you listening?
I think the key thing is that machine learning, like these random forest models, really does show promise for, well, decoding this incredibly complex plant microbe dialogue.
And that Actinomycetales finding is a really strong lead to follow up on.
Definitely. It points towards potentially key regulators within the microbiome.
And the broader implications for agriculture seem huge.
Oh, absolutely. If we can reliably predict and then design these interactions, it opens the door to tailored microbial products.
Which could mean, better crop yields, maybe using fewer chemicals, more sustainable practices overall.
That's the hope. More sustainable, resilient agriculture.
So here's something for you to think about. We've talked about understanding and predicting, but what if we could take it further? What if we could precisely engineer a plant's microbiome? Could we create crops that are naturally more productive? Yes, but also inherently resilient to drought, heat, disease, things linked to climate change, reducing our reliance on conventional fertilizers and pesticides. Imagine agriculture transformed by harnessing this hidden world beneath our feet.